This is just a quick little bit of extra information to help you with the chemical oceanography practical. So there's a part of it that's about marine sediments, and the idea is to give you guys some experience at looking at a real archive of Earth history by showing you core photos and smear slide data from actual cores that have been collected in the marine environment. And so the goal is for you to take a look and describe the physical characteristics of the sediment cores, use the smear slide data to identify some of the main sediment components and where they come from, determine the lithologic names of the marine sediments using the handout, and then eventually in class I'll make a map and show you guys some of the distributions of the primary marine sediments. Okay, so just a couple of practical things to get you started. So this photo on the right here should look familiar because these are some of the photos of the example cores I gave you. This is similar. And they have these complicated names with all these numbers and letters and all these things. So just to give you a sense. So the first number is the leg or the expedition. So in this case, it would be leg 199. And then the site number is site 1215. So you can see here, these are the sort of things. So imagine the Joy User Resolution, the big coring ship goes out and the number of the expedition is 199. And you've been on the cruise and you've done a bunch of sites, but now you're at site 1215. That's the location where you're at. And then you've got hole A in this case, because you're going to do more than one. Core 2H. So you can see here the core sections are numbered. So you've got your water depth and then you hit the sediment. And then you've got from here, this is the sea floor, and this is how deep your core is. And so you've got the first part, the second part, and then the third part. So this particular example here is core 2H, which means it comes from this part. And then section 5. So now you're starting to look at sections here. So this is section 5 is the part that you're looking at. So if you look here at core 2H, you've got now this blow up of it. So core 2H now is split into various sections. We've got section 1, and then there's a little missing bit, and then section 2, 3, 4, and now section 5 is the one that we're particularly looking at. And then of section 5, it's from 0 to 150 centimeters. And so this is section 5 here, and so the section that you've been given, quote unquote given, or the sample that you've been given is from right here, 80 to 85 centimeters, and you can look here on your little thing. So it's essentially this piece is what you've been given if you've been given 199-125A-2H-5, 80 to 85. Okay, so that just gives you a sense of how this works. You can see that it sort of makes sense. So core, this 2H that you see in the photo is sort of from the middle, but you can see even throughout that 2H, there's differences in the type of material you're encountering. The color's different, the packing is different, etc. Okay, so here are just, you've got some examples of different cores. They look, really incredibly different when you think about it in terms of the color. So these are cores from different expeditions. You can see they've got completely different leg numbers, like 130, 145, 206. Some of them have fractures and things. There's different layers, all sorts of stuff. So these cores, you can get a lot of information, and they're quite dramatically different from different parts of the seafloor. The other thing to note is that a lot of times, so these photos will be taken immediately. The cores will come up and they'll be split and then cut in half. And as a scientist, a lot of times you're only presented with these core photos and you have to choose, what part of it you'd like to study. Because obviously it'd be great if you could get your hands on material from everywhere, but you can't, right? You have to share with the whole scientific community. Okay, So one thing that the scientists on ship will do in order to assist the scientists elsewhere who will be working with the cores is they'll start with these photos and generate as much information as they can by creating a barrel sheet. And so if you look on the left, you can see it has some identifying information about meters and section and where it is. At the top, you've got the name here. And then what they're using throughout is they're essentially, if you look here through this sort of lithology section with this blue and green and yellow, they're using symbols to describe what it is they're seeing throughout the core. And I'll show you next what the sort of symbol things suggest. So a couple of interesting things. They've got this little section here, these little gray bits where they're marking where there's evidence of bioturbation. And then they're also showing where they see structural features down the core. So they're using these sort of symbols to show that here. And then under sample, they're also showing where they took a smear slide sample. So you can see here all the little SS's means they took a little slide for microscope investigation, whereas the IW means they took some of the interstitial water, so they got as much liquid out as they could. And then if there was a PAL, it means they took a sample for micropaleontology. And then the color, of course, they're indicating what color the sediment looked like, what color was the mud. And then on the right, they're giving as much description as they can. And so these little symbols, the little blues and greens and yellows that you're seeing here, come from a key like this. So if you see siliclastic sediments such as sand or sandstone, you would use this to color it in. And then if you had diatom ooze, you'd use this. And then to indicate various types of geological or bioturbation activity, you use these various symbols. And you can see on the right all the different core disturbance symbols, the bioturbation intensity, all these various sedimentary structure things. Now, obviously, a lot of this, this is the real sort of sheet that scientists would use, and it's way more in-depth and detailed than I would ever expect you guys to go into. But there's certainly some of this you can recognize. And so if you were looking here, you could obviously get the color right. You could get that there was some kind of bioturbation. Here you can see, for example, there's clearly some sort of a difference happening here between one type of sediment and another. And you can also use the smear slide data you have to help identify where the various features are. So this one is a combination of nanofossil ooze, and then the green lines mean the nanofossil ooze is mixed in with silty clay or claystone. Okay, so just have fun with the barrel sheet. It's not supposed to be a super intense thing, it's just supposed to get you introduced to some of these ideas and things. And again, the last thing I'll point out, because these are real cores, you guys can go ahead and sleuth around the internet and find as much information as you can if you want to. You don't have to, but the studies have been done on the various cores I presented you guys, and I intentionally chose a couple that were really different from one another. Okay, so last thing is for these smear slides, I wanted to show you guys some photos from under the microscope of what these things actually look like. So this is, you can see quite small. The scale bar here is 0.05 millimeters. This is what volcanic glass looks like under the microscope. And then this is a feldspar mineral surrounded by volcanic glass. So again, just some cool examples of what these smear slides look like so you can get a sense for the cores you're looking at. Here's silt minerals and some mica flakes. Clay. Not as exciting looking, but clay with some dolomite. And then here, this is where it starts to get a little more interesting because now you're seeing some biological examples. So here you've got some siliceous clay, and you've got some centric diatom fragments, and some silica flagellates. That's your silica flagellates here. And this you would consider your siliceous ooze. Basically, all of these sort of glass rod-looking things you're looking for are frustules or sponge spicules that have broken off organisms, and you can see the various types of shells that have broken apart all throughout here. These are nice and intact sponge spicules, so these are siliceous sponge spicules. They're actually quite big. And then here, the much smaller, are the calcareous nanofossils, so lots of little coccolith plates Have to use a special kind of polarized light to see them, but these are all those little coccolith 4 plates everywhere. And then these are examples of what forams look like underneath. You can see this is a virtually intact foram shell, which is quite cool. And you can see some smaller ones here. They look like these little blobs. And then that's it. So then the other thing I just wanted to show you real quick was if you go to joydesresolution.org, it's a pretty cool website if you want to spend a couple of minutes. Talks about the ship that's used for this type of coring, get some information about the ship, where it's been, and you can look at, here would be a good place to find some of the past expeditions for some of the cores that I've given you guys. You can go on for the public or for educators. You'll see there's some really cool stuff. I think you can just learn about how the cores are